Hello friends, my name is Rizwan Hafeez and today we are going to talk about a very important concept, that is difference between genes and alleles. If you think that it is difficult to understand the difference between genes and alleles, stay with me just for 5 minutes. I assure you that at the end of this lecture you will be very much clear about genes, alleles, homozygous and heterozygous organism. Okay, now let's start. So as we know that, for example, this is a human cell, and in our human cells there are basically, we know that 46 chromosomes. There are 46 chromosomes. We know that 23 chromosomes come from the mother side and 23 chromosomes comes from the father side. Okay, there are basically 46 chromosomes. Okay, these 23 chromosomes coming from mother side, for example 20, they are n number of chromosomes. Okay, and similarly 23 chromosomes coming from father side, they are also n, haploid. Okay, now 23 plus 23 will make 46, and similarly over cell n plus n, it will be 2n. N plus n, it will be 2n. Clear? Now the chromosome number 1 from the mother side and the chromosome number 1 from the father side, they both are basically homologous chromosomes. They will be called as homologous chromosomes. Similarly, chromosome number 5 from the mother and chromosome number 5 from the father, they are homologous to each other. They are homologous chromosomes. Chromosome number 7 from the mother, chromosome number 7 from the father, they are homologous chromosomes. Chromosome number 20 from the mother and 20 from the father, they are basically homologous chromosomes. So now you are very much clear about what are homologous chromosomes. What are homologous chromosomes? Now let's, for example, this is chromosome number, let's suppose 1 from the mother side and chromosome number 1 from the father side. Okay, so it means if this is chromosome number 1 from the mother side, this is chromosome number 1 from the father side, so it means both of these chromosomes are homologous chromosomes. Homologous chromosome. So we know that chromosome do have DNA in it. Okay, in the next video I will explain about what is difference between chromatin and what is difference between chromatin and chromosomes, how chromosomes are formed. In the next video I will explain about it. So this is a double-stranded DNA present inside the chromosome, and here is a double-stranded DNA present inside the chromosome. We know that chromosomes do have genes. So to understand the concept of genes, first of all let me elaborate a little bit about the structure of DNA. In the previous videos I have explained about detailed structure, how nucleotides get attached, what type of bonding is being formed, and how the detailed double helix structure of DNA is being formed. All these things has been explained in my previous video. Here I will give you a very brief summary. So we know that DNA is made up of nucleotides: adenosine, thymidine, guanosine, cytidine. Different types of nucleotide, they attach with each other and a double helix DNA structure formed. Okay? So for example, adenosine will get attached with the thymidine, and thymidine will get attached with the guanosine, and guanosine will get attached with the cytidine through phosphodiester bond, and similarly again C, again G, for example again C, again A, and similarly in the complementary strand against A there will be thymidine and double bond will be formed. Against T there will be adenosine, double bond will be formed. Against G, guanosine, there will be cytidine, triple bond will be formed. Against C there will be G, double bond will be formed. Against C there will be G, double bond will be formed. Against G there will be C, triple bond will be formed. Against C there will be G, triple bond will be formed, and against A there will be T and double bond will be. In this way a long double helix DNA structure will be formed. Okay, so DNA is basically polymer of these nucleotide. Okay, now so what is gene? On the DNA, double stranded DNA, the sequence of nucleotide, sequence of few nucleotide, 100 to 1000 nucleotides, okay, that code for one particular protein, that protein might be for the color of the eye, that protein might be for the color of hair, that protein might be for some particular enzyme. Okay, now for example this is a gene. Now you know what is transcription? Transcription is basically conversion of DNA into RNA. So it's not like that whole DNA will be converted into full-fledged RNA. No, this is not possible. One gene will be converted into one RNA, and this process is called as transcription. Now this RNA, it will go out of the nucleus, it will attach on the ribosome, and one protein will be formed, and that protein might code some specific phenotype like color of eye or color of hair or anything else. Okay, so it means one gene will code for one particular protein. The process of conversion of gene into RNA is transcription, and the process of conversion of RNA into protein is called as translation. So now you are clear what is gene basically. Okay, so on the DNA of these homologous chromosomes there will be lots of genes present. For example, this is one gene, sequence of nucleotide. This is one gene, 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 and similarly on this chromosome that is coming from the father side, this is one gene, this is one gene, and similarly one gene and one gene. Okay, now these are genes. Now what are alleles? Now alleles are the genes present on homologous chromosome at same position, at same locus. So here you can see this gene is present at the same locus, at the same position at which this gene is present. So the genes present on homologous chromosome on the same locus, same position, they are called as alleles of each other. They are called as alleles of each other. So both these genes are basically called as alleles. The genes present on homologous chromosome on the same locus, same position will be called as alleles of each other. So these are alleles. Similarly, these genes, they are present on the same locus, they are called as alleles. And similarly all these things, all these genes present on the same locus, they will be called as alleles of each other. Clear? Okay, now assume that these genes are basically responsible for the color of eye. The protein from these genes that will be produced after the process of transcription and translation will regulate the color of eye. Okay. For example, this gene, the present on this homologous chromosome, will code for brown color eyes, and this gene present on this homologous chromosome will code for blue color eye. Okay, blue color eye. This gene code for brown color eye, and this gene code for blue color eye. Now, both the genes are different. They are present on the same locus, but they are different genes. So this type of organism will be called as heterozygous. If both the alleles present on the homologous chromosome are different, the organism will be heterozygous. Heterozygous. See, but for example, if this gene code for brown color and this gene also code for brown color, now both the genes present on homologous chromosome are same, now the organism will be called as homozygous. Homozygous. If the genes are different, for example, brown, this gene code for brown color and this gene code for blue color, the organism will be heterozygous. Heterozygous. So heterozygous and homozygous. I think now you are very much clear about what are homologous chromosome, what are gene, what are alleles, and what are heterozygous and homozygous organism. Please don't forget to like my video and share my video and subscribe my channel and press the bell icon so that you may get notification for the future videos. In the upcoming videos, I will be explaining about the difference between chromatin and chromosome. I will also explain the difference between chromatin and chromatid. Okay, how chromosomes are formed. All these things will be explained in my upcoming video. So please don't forget to press the bell icon so that you may get notification. Thank you so much.